Greetings, everyone, and welcome to today's webinar, Key Financial Viability Considerations for Laboratorians Considering Bringing Clinical Pharmacogenomics Testing into the Laboratory. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, visit thermofisher.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. Finally, as a reminder, today's presentation is educational and offers free continuing education credits. Simply click on the Continuing Education Credits image located at the bottom of your screen and follow the process to obtain your credits. I would now like to welcome our speaker, Becky Winslow, founder inGENEious RX. For a complete biography on Becky, please visit the Biography tab at the top of your screen. Becky, welcome. You may now begin your presentation.
Thank you for joining me today as I present Key Financial Viability Considerations for Laboratorians Considering Bringing Clinical Pharmacogenomic Testing into the Laboratory. My name is Dr. Becky Winslow. I'm the founder and head consultant for inGENEious RX, a boutique firm specializing in bench-to-bedside pharmacogenomics consulting. I'm an authority in pharmacogenomics access and reimbursement, and I specialize in business strategy consulting for pharmacogenomics stakeholders. Before I go any further, I need to credit Dr. Mary Wiseman for assisting me with creating the webinar slides and Dr. Kristen Spear for creating the original version of this inGENEious RX PowerPoint template. With that said, let's dive into the webinar. Laboratories may use the same equipment they use for COVID testing to run pharmacogenomics testing. With COVID testing reimbursement dropping, many laboratorians are considering implementing PGx testing. By the end of today's webinar, my goal is for you to make educated financial decisions when considering implementing clinical PGx testing in the laboratory, to differentiate between implementing a clinical PGx test for which CPT codes and payer policies exist and implementing one for which they do not exist. And thirdly, to describe how to optimize PGx test panel design and utilization using payers' existing coverage and reimbursement guidelines to positively impact the PGx laboratory's profits. By considering a patient's genetic profile, pharmacogenomics seeks to optimize drug therapy and improve drug therapy outcomes. Pharmacogenomics clinical adoption and pharmacogenomics laboratories' financial viability and sustainability depends on its market access and reimbursement. Based on what you know right now about PGx's market access and reimbursement, how would you vote if I told you I'm going to build the most comprehensive PGx test in the industry and all payers will buy it? And by most comprehensive, I mean a multi-gene drug test that covers multiple disease states. Would you vote, Becky is well informed about the PGx payer market and I want to learn how to build the same? Or would you vote, Becky is misinformed about the PGx payer market and I'm not following her lead? Or would you vote, I'm uncertain if she is well informed or misinformed, but I need to know whether to follow or disregard her? The fact is, as a laboratory stakeholder considering implementing PGx testing in the laboratory, you need to know the correct answer to this poll. And by the end of this webinar, you'll have the information you need to do so. Many want to implement PGx testing, some into existing laboratories, and some want to open a new laboratory. So what do you need to consider to make an educated decision? You've seen all the impressive numbers about PGx that suggest a PGx test would benefit everyone, such as 99% of the population has at least one genetic variant that affects their response to medications. And you've probably also heard that over 300 FDA-approved medications have PGx references in their labeling. This data may seem promising, and you may think you should create the most comprehensive PGx test on the market, so as many people as possible can benefit from the same test, and your laboratory can generate the most revenue per test. However, before you do that, you need to think about a few things that I'm going to cover with you in this webinar. You may be considering, do payers currently cover and reimburse all PGx testing? How many drug gene pairs do payers currently reimburse? And what are payers' utilization guidelines? And you may also be wondering, what about those drug gene pairs that are yet to be discovered, or they're discovered but don't have CPT billing codes and payer coverage and reimbursement policies? The answers to these questions are critical to your success. So let's dive into the answers. We will start by reviewing laboratory-developed test implementation. As we transition into this section of the webinar, I encourage you to start thinking about the end, keeping the end in mind. And by keeping the end in mind, I mean, let's look at the smart financial decisions you're going to have to make sure that your lab can meet its mission. Before implementing PGx testing, laboratorians should consider all steps of the implementation process, just as they do for all other laboratory-developed tests. The 6 steps in the implementation process include lab setup and accreditation, planning, test configuration, analytical and clinical validation, and launch. Key considerations for a PGx lab's viability include: why implement a PGx test? What drug gene pairs will the test or panel include? Is the test set up properly and performing as expected? Are test results clinically accurate? And what are the best practices for introducing the PGx tests? While the lab accreditation and setup step is important for PGx's financial viability, most labs master this step, so we will focus specifically on the steps I've witnessed labs struggle with the most over the last decade. And those are the planning, test configuration, and launch steps. I will group the analytical and clinical validation steps under the configuration step. Missteps in implementing the right PGx tests and allowing test users to misutilize a test have been costly for many PGx labs over the years, and for some, it's resulted in their demise. For the planning step of implementation, we will focus on reimbursement, because accurately estimating PGx reimbursement is critical to calculating the potential return on your investment into PGx. When a laboratory begins planning a PGx test, it should carefully consider who its potential payers are, if billing codes and clinical utility evidence exist, and if the payers have coverage and reimbursement policies for the billing codes, or if the lab must request new billing codes, prove its test clinical utility, and petition payers for coverage and reimbursement. Why must this be a priority? Because payers require clinical utility evidence, and coverage is tied to coding. These factors influence how soon the lab will receive reimbursement after a PGx test is implemented. So we will examine the LDT reimbursement pathway, in which billing codes, clinical utility evidence, and payers' coverage and reimbursement exist first. And just to clarify before we move forward, the billing codes I'm referring to are International Classification of Diseases 10th Revision, or ICD-10 codes, and Current Procedural Terminology codes, also known as CPT codes. I also want to specify that CPT codes include Proprietary Laboratory Analysis codes, or PLA for short. In this LDT reimbursement pathway, payers have defined the right test panel design and the right utilization of the test. The payer has defined the right test by drugs and genes, or CPT codes, and the right utilization by the right patient, or ICD-10 codes, the right prescriber, the right time to test, the right billing and coding, the right documentation to support medical necessity, and the right reimbursement for the test. The lab simply needs to identify the payers and their definitions of the right test and the right utilization, and follow their definitions when implementing their PGx tests. The researcher will find these in payers' coverage and reimbursement and billing and coding policies. And I'm going to share examples of payer policies with you later in this webinar. So just to recap, when CPT codes exist and robust evidence exists proving the test's clinical utility in the intended population, these codes are covered under the radar and they require minimal effort for your laboratory to implement. They also generate revenue faster. In this LDT reimbursement pathway, payers have not defined the right test panel design, nor have they defined the right utilization of the test. The payer has not defined the right test by the drugs and genes, and the right utilization by the right patient, the right prescriber, the right time to test, the right billing and coding, the right documentation to support medical necessity, and the right reimbursement for that test, as I mentioned previously, when there are existing codes and evidence. In short, payers have not defined their coverage and reimbursement guidelines. So this is unknown to you. What do they see as the right test? And what do they see as the right utilization? And this is important to you because obtaining reimbursement for pharmacogenomics testing requires multiple steps. First, the lab must prove the test's clinical validity to the American Medical Association, who will issue a CPT code when they are satisfied that the test is clinically valid. Once the lab obtains a CPT code, the lab then must establish the monetary value for the test. Then payers decide if they will reimburse the test. And payers require robust clinical utility evidence, as I've mentioned before. Most labs who pursue this LDT reimbursement pathway partner directly with payers to identify adverse drug events and or non-therapeutic drug outcomes in the payer's patient population, and then they build the PGx test and the clinical utility data the payer desires in parallel and in collaboration with the payer. This as opposed to building a test the lab thinks will solve the payer's drug problems, and then later discovering that the payer doesn't view those drug problems as a big enough problem for them to invest in solving it. So just to recap, when a lab must request new CPT codes from AMA, and the lab must prove the test clinical utility in the payer's population to earn coverage and reimbursement, these codes are not going to be covered under the radar. So this process is going to require more time and financial investment from your laboratory. You're also going to generate revenue slower. However, it is important to note that minimal risk exists once payers award reimbursement. And by minimal risk, I mean that the payer is less likely to deny reimbursement when you bill for your test. So let's summarize the two pathways to LDT reimbursement before we move on. And let's summarize them in terms of the time from test implementation to reimbursement. One is the turtle, and one is the hare. For the remainder of this webinar, I will focus on covered under the radar PGx testing, or specifically how laboratories may optimize PGx test panel design and utilization using payers' existing coverage and reimbursement guidelines for pharmacogenetic associations that are related to drug metabolizing enzyme gene variants, drug transporter gene variants, and gene variants that are related to a predisposition for certain adverse events and drug efficacy. So far, we have reviewed the planning step in the implementation process. The takeaway was to start with the end in mind by understanding reimbursement pathways. Now we will focus on the test configuration step in the implementation process. To master this step, you must know the payer landscape. You must map payers' policies and design the right test as the payer defines it. So let's dive into the payer landscape in the U.S. PGx testing insurance coverage in the U.S. has improved over the last 5 years, but still varies widely. Some labs operate on a cash basis due to coverage limitations. In this webinar, we're focusing on non-cash payers because an estimated 304 million, or 92.1% of Americans, had health insurance at some point in 2022, which is slightly higher than that of statistics for 2021, and that's according to the U.S. Census Bureau. The most common health insurance in the U.S. is employer-based, followed by government-provided public health coverage such as Medicare and Medicaid, and private non-group health coverage. Medicare's decisions influence PGx reimbursement and set a baseline for private insurers. Coverage for specific testing varies among insurers. A payer's reimbursement decisions are influenced by factors such as clinical utility, which we mentioned earlier, cost effectiveness, evidence quality and quantity, professional guidelines that exist for the test, and regulatory requirements and medical policies. As I mentioned earlier, payers define the parameters for the right test. They define the right patient, the right prescriber, the right time for the test, the right billing and coding, the right documentation, and lastly, the right reimbursement. Because understanding payer policies and billing and coding guidelines is so crucial for a lab when configuring tests, I will now show you a closer look at each non-cash payer. Let's focus on Medicare first, because non-Medicare payers model their policies upon Medicare policies. CMS reimburses laboratory tests based on the clinical laboratory fee schedule. The clinical laboratory fee schedule defines the specific dollar reimbursement rates for each type of test and applies to all Medicare administrative contractors. Medicare uses MACs to administer the Medicare program, such as Medicare Part A and Part B fee-for-service medical claims for PGx testing. Looking at this map, you can determine which MAC administers your PGx. Most MACs subscribe to the Molecular Diagnostic Service Program, otherwise known as MolDX. The program's purpose is to provide uniform policies for 28 states across the CGS Noridian, Palmetto, GBA, and WPS Max. I will explain why this is relevant on the next slide. Medicare mandates national coverage decisions at the national level, and all MACs follow those guidelines. Only one NCD currently exists for PGx testing, and that is the NCD for pharmacogenomics testing for warfarin response. The NCD limits coverage of PGx testing for warfarin response to coverage with evidence development. In other words, Medicare does not believe sufficient clinical utility evidence exists to reimburse PGx testing of CYP2C9 or VCORC1 alleles to predict warfarin responsiveness for their beneficiaries. But they will cover the testing for a controlled study that attempts to prove the clinical utility of the testing. MACs use local coverage determinations to determine whether they will cover a particular service on a MAC-wide basis, and their PGx LCDs may vary, except for the MACs who subscribe to the Molecular Diagnostic Service Program that I mentioned previously. Knowing payers' special coverage determinations, such as Medicare's SCD for CYP2C9 or VCORC1 alleles, or MolDX's requirement for a technical assessment is vital for laboratories when configuring their test panel and allowing users to utilize the test billed to the payer with SCDs. In the NCD example, unless a lab plans to conduct a prospective randomized controlled clinical utility study for CYP2C9 and VCORC1 for warfarin, no MAC will reimburse those genes for warfarin. Likewise, MolDX will not reimburse a test for which a lab hasn't passed its technical assessment. Managed care is a dominant delivery system for Medicaid enrollees, with 72% of Medicaid beneficiaries enrolled in comprehensive managed care organizations nationally. As of 2020, more states use Centene as their MCO than any other MCO. Centene subscribes to Consortium's PGx policy. Consortium is a team of clinicians, researchers, data scientists, and software engineers who author genetic test policies for health plans. Consort's PGx policy is restrictive as it deems medically necessary single gene drug PGx tests, such as CYP2C19 and clopidogrel, but deems multi-gene PGx panels investigative and non-medically necessary. With that said, when state Medicaid coverage provisions conflict with the coverage provisions in Consort's policy, state Medicaid coverage provisions take precedence. This may happen when a state legislates that all PGx testing will be covered, such as in the case of California. According to the U.S. Census Bureau, more than half of the people in the United States have health insurance through their employers, which is the most prevalent type of health coverage in the country. Blue Cross and Blue Shield, United Healthcare, Cigna, Aetna, and Humana, commonly referred to as BUCA, are the largest providers of employer-sponsored healthcare in the U.S. per capita. Their policies differ in comprehensiveness and clarity, to be honest. United Healthcare, for example, deems medically necessary multi-gene panels, up to 15 genes, to guide therapy decisions for antidepressant and antipsychotic medications when all the following criteria are met. The individual has a diagnosis of major depressive disorder or generalized anxiety disorder, and the individual has failed at least one prior medication to treat their condition. United doesn't, however, deem medically necessary panels with 5 or more genes for genetic polymorphisms for any other indication. Cigna, in contrast, doesn't cover any multi-gene panels, no matter how small the panel or the patient's diagnosis. Cigna states language early in its policy that might lead one to believe their policy has comprehensive coverage. Yet later in the policy, they limit coverage to only 6 CPT codes associated with a very small subset of medications and diseases, while claiming insufficient evidence exists to support the clinical utility of PGx testing, for which, quite frankly, many other payers reimburse. For example, Cigna covers G6PD testing, but doesn't cover CYP2C19 testing that most payers do reimburse. I bring these examples up to show you that payers' PGx policies can be confusing and seem inconsistent in and of themselves, and when compared to other payers' policies, they may seem even more perplexing, even to the expert. Now that I've outlined the majority of PGx payers in the US, I will enlighten you on how payers define the right test and the right test utilization. We will start with the right test configuration, as it serves the basis for all other steps in the PGx test implementation process after labs identify payers in the planning step. While I did not mention this when discussing different payer policies earlier, the criteria all payers share when defining the right test is that the test is analytically valid, and clinically useful. Analytical validity refers to a test's ability to measure the genotype of interest accurately and reliably. Clinical validity refers to a test's ability to detect or predict the clinical disorder or phenotype associated with the genotype. The clinical utility of a test is a measure of its usefulness in the clinic and resulting changes in clinical endpoints. Payers start to differ in their definition of the right test when it comes to the genes and drugs included in the right test. So just to recap, the right test is defined by the right genes and the right drugs, and each payer has different definitions. The most common drug gene payers agree to reimburse are those for which the drug's FDA label states testing is necessary for therapeutic recommendations or to prevent drug toxicity, and also the Clinical Pharmacogenetics Implementation Consortium's Level A and B drug pairs. And here are some examples of those gene and drug pairs on the left-hand side of the screen. Thus far, we have discussed the planning and test configuration steps in the PGx test implementation process in depth. Let's wrap up by discussing the last step, which is the launch step, and specifically the right test utilization, as it is equally important to the lab's financial viability. My clients often ask me if stratifying patient populations to identify patients for which PGx testing is medically necessary by the presence of polypharmacy, patients' ages, the facility type, patients that have experienced therapeutic failure or adverse drug events, and or patients who have a family history of therapeutic failure or adverse drug events is a good technique. My answer is yes, for patient stratification from the 100,000-foot view of a population. However, the patients by that criteria still may not qualify for testing at the individual payer level. As I pointed out previously in this webinar, payers are very specific about what PGx testing they pay for. And again, they define the right test and they define the right patient for that test. The right patient is defined by the patient's ICD-10 codes. We said earlier that ICD-10 codes stand for the patient's diseases or conditions, such as major depressive disorder or generalized anxiety disorder. Also, payers define the right patient by the drugs that they are taking. So if a patient isn't consuming a drug that they deem to have PGx relevance, then they do not deem necessary a PGx test for that drug. So it's important to put all of the pieces of the puzzle together. We need ICD-10 codes. We need drugs that are on the test to identify the right patient. MolDX defines the right prescriber clearly and concisely, so I will use their policy as an example. MolDX specifically requires the ordering PGx test provider to be a provider who has the licensure, qualifications, and necessary experience or training to both diagnose the patient's condition and prescribe medications for the pharmacologic management of the patient's conditions. Secondly, the prescriber must be considering or has already prescribed a drug for which actionable drug-gene interactions exist, and only those Medicare recognizes as medically necessary. The clinician must also have made an initial personalized drug decision for the patient based on all non-genetic factors that go into the medication prescribing decision-making process, such as the patient's medical history, their other medical conditions, other medications the patient's taking, as well as basic science pertinent to the drug, such as regular drug-drug interactions. MolDX also specifies that the ordering prescriber must understand the actionability of the tests they order. These prescribers are not as common as you may think they are, based on my experience over the last 10 years in PGx. And the reason for that is because clinical PGx is still a novel topic that is in med schools and even in pharmacy schools. And its implementation is still a novel process for many prescribers. And this is why your lab must be diligent in educating potential prescribers about its PGx tests and their implementation. If the lab is to be successful over the long term, the lab must also commit to not bill tests that non-qualified prescribers order. Processes exist for labs to protect themselves, and we can talk about these in another webinar or after the webinar. Your prescriber's understanding the right time to test is critical, and the patient's medical records should reflect that the prescriber ordered the PGx test at the time the payer deems medically necessary to avoid financial clawbacks should the payer audit the patient's medical records. Payers may approve PGx testing prior to the initiation of a drug, but many only approve it post-initiation when the patient has already experienced an adverse drug event or a therapeutic failure. The first example I provide here is Humana's policy for CYP2C9 genotyping prior to the patient beginning Mayzent for the treatment of multiple sclerosis. In this case, Humana would not deem medically necessary testing the multiple sclerosis patient after they have already started Mayzent, and upon audit, the payer could recoup the reimbursement for that test. The second example I provide is UnitedHealthcare's policy for prescribing a multi-gene panel to guide the prescriber's antidepressant and antipsychotic medication choice after the patient has failed at least one prior medication to treat their major depressive disorder or generalized anxiety disorder. Should a prescriber order the test prior to the patient failing at least one prior medication, UnitedHealthcare could recoup the reimbursement upon auditing the patient's medical record. So just to summarize, labs must be diligent in only billing PGx tests that prescribers order at the right time per the payer's utilization policies. Each payer may have special documentation requirements also, and labs must be persistent in ensuring prescribers follow them to protect their reimbursements. Again, using MolDX as an example, MolDX requires the prescriber to document clearly in the patient's medical record that the patient has a diagnosis for which pharmacologic therapy is reasonable and necessary. They must also document in the medical record the drug or drugs that the clinician is considering using, and that they are reasonable and necessary for the treatment of the patient's diagnosis. Again, those drugs need to be drugs that are on the right test as we defined it earlier in the webinar. Thirdly, the prescriber needs to document in the medical record that they did choose the initial drug based on non-genetic factors. I talked about this in the previous slide when we talked about the right time to test. It's equally as important for the prescriber to document that in the patient's medical record. The prescriber must also document what drugs the prescriber is considering as alternatives to the initial drug. Again, those drugs either need to be on the right test or they need to be alternatives to the right test. The prescriber must also document the medication action plan detailing how the prescriber will use the PGx test results and in what time frame the prescriber will use them. I personally witnessed Medicare recoup millions of dollars in PGx test reimbursements from a lab who billed PGx tests for which the prescribers had not documented the patient's charts as the payer defined. I know as a lab you may be thinking, how can I protect my lab from payers recouping payments when I can't control what the prescriber documents in the patient's medical records? And we often don't have access to the patient's medical records to verify proper documentation. Solutions exist beyond the scope of today's webinar. You can ask me after the webinar or contact me separately to discuss those options. As I said earlier, payers also define the right billing and coding. The CPT coding system is used to code medical services and procedures, including pathology and laboratory tests that encompass pharmacogenomics testing. Tier 1 codes apply to the more common gene variants and contain the most information, including the analysis specifics. Analysis specifics are important when considering the method by which you will perform PGx testing, because payers require labs to use the CPT code that most accurately describes the service being rendered. Payers are most likely to reimburse Tier 1 codes. Tier 2 codes are characterized into 9 levels of complexity, and they represent less common and lower volume PGx assays. Payers are less likely to reimburse those, although Cigna, for example, who has a narrow PGx coverage reimbursement policy, does reimburse 81400, 81401, and 81405, which are Tier 2 codes. Likewise, most payers do not reimburse 81479, which is the CPT code for an unlisted molecular pathology procedure. So the take-home message is that the payer does define the right billing and coding for the right genes in their coverage and reimbursement policies. When configuring your test panel, know what CPT codes payers will and won't reimburse and how much they reimburse to make smart financial decisions. I mentioned the MolDX MAC earlier and the states that are included in that MAC and how they have uniform policies. Laboratories in the MolDX MAC must register with the DEX Diagnostics Exchange that Palmetto GBA designed to identify molecular diagnostic tests and manage policies for the tests. Its purpose is to connect payers and labs by clarifying to payers exactly what tests the lab is submitting for reimbursement and clarifying to labs exactly what the payer will reimburse for the tests. MDX tests are uniquely identified and cataloged using DEX Z-code identifiers. Palmetto's, which is Palmetto GBA's unique and proprietary 5-character alphanumeric codes assigned within the DEX Diagnostics Exchange. So once you have your right test, the lab must register in the DEX Exchange. It must register the lab's test in the DEX Exchange. DEX will assign a Z-code to the test. The lab submits a technical assessment for the Z-code. And DEX determines coverage and reimbursement for Z-codes. And laboratories use those Z-codes on their billing forms when they submit the test for payment. So let's look at the last important factor, the right reimbursement. Payers also define the right reimbursement just as they do billing and coding. The lab must be meticulous in ensuring payers reimburse them the maximum reimbursable amount for their tests. Labs can enact special procedures to do so. Prior authorization and pre-claim review are two such procedures that are similar, but differ in the timing of the review and when services can begin. Under prior authorization, the provider or supplier submits the prior authorization request and receives a decision before services are rendered. Under pre-claim review, the provider or supplier submits the pre-claim review request and receives a decision prior to claim submission. However, the provider or supplier can render services before submitting the request. Also critical to your lab's financial stability is verifying the reimbursement balance to make sure it's correct and following up immediately with the insurer for clarification when discrepancies arise. So let's recap the webinar thus far. We are looking here at the pathway that will get you to reimbursement the fastest and with the least effort on your laboratory's part. As we've mentioned, the payer has already defined the test panel. They've already defined the utilization. And your laboratory simply needs to identify those payers, their definitions of what the right test is, and the right utilization, and follow their definitions. As I mentioned earlier, these are found in their billing and coding policies, and I showed you examples of where you can find them. So to summarize our webinar today, making educated financial decisions when considering implementing clinical PGx testing in the laboratory is critical for laboratories' viability and sustainability. And in terms of time from test implementation to reimbursement, implementing a clinical PGx test for which CPT codes and payers' policies exist is faster than implementing one for which CPT codes and payer policies do not yet exist. Payers define the right test, the right billing and coding, the right patient, the right prescriber, the right time to test, and the right reimbursement in their coverage and reimbursement policies. And lastly, optimizing PGx test panel configuration and utilization according to payers' coverage and reimbursement guidelines can positively impact the PGx laboratories' profits and losses. So let's revisit the poll that I asked you at the beginning of the webinar. How would you vote if I told you I'm going to build the most comprehensive PGx test in the industry and all payers will buy it? Let's see if your answers are the same. The first choice is Becky is well informed about the PGx payer market, and I want to learn how to build the same comprehensive test. Would you vote Becky is misinformed about the PGx payer market, and I'm not following her lead? Or lastly, would you vote still that you're uncertain about whether I'm well informed or misinformed, but you want to know the type of test to build that payers will reimburse? I hope that I was able to show you that I'm well informed about market access and reimbursement for PGx during this webinar, and also that I've equipped you with the information that your laboratory needs to get started with knowing the right type of test to build that payers will reimburse. Thank you for joining us for the webinar today, and I'm now going to open the floor for questions.
Becky, thank you so much for that excellent presentation for our live Q&A portion of our webinar. So I do want to remind our audience, although we have quite a few questions already that have come in, if you have any questions and you haven't submitted them, please do so now. Type them into the Ask a Question box and click Send. Or actually click Submit, and we will answer as many of your questions as we have time for. And here we go. Becky, I'm going to dive in right now. Let's start with this one. How long does it take from enrolling in the DEX exchange until the lab is able to bill molecular diagnostics for their PGx testing using their Z code?
Great question. And also very important to your lab's financial viability. The process from enrolling in the DEX exchange to being able to bill MolDX for PGx tests involves several steps, as we mentioned, and it can take approximately 60 to 90 days. Of course, there's different variables that go into how long it takes. For example, your laboratory should have their technical assessment prepared before you register for a Z code. If you do not have your technical assessment prepared, then that 60 to 90 days is going to turn into way more than 90 days. And after you've received the email from enrolling in the DEX exchange, you'll have access to add your test information. So we're looking at about 90 days if all your cards fall in place and it goes exactly as it's planned to go.
Thank you, Becky. Our next question, this one's two parts. Have any laboratories succeeded in earning reimbursement for their PGx test after completing their MolDX technical assessment? And if I make changes to my PGx test, do I have to submit for another Z code?
Those are also great questions, and one that I get quite often. So yes, several labs in the MolDX MAC have successfully completed their technical assessment and they've earned reimbursement. Those reimbursements are averaging around $917. But with that being said, you need to know that different variations of PGx tests have passed those technical assessments and have earned reimbursement. So important to consider that your lab's test may be a little different, but it looks like as PGx is being recognized for its clinical utility, more labs are passing their technical assessments and getting reimbursement. Now, as far as if you change your test, do you have to submit for another Z code? Again, that's another great question. No, you do not have to submit for a new Z code if you make changes to a PGx test. Instead, you should log into the DEX exchange and update the existing test record with the new information. However, you do need to note that all components of the panel must be registered. So if there's a difference in the method used to acquire the results in your panel, then an application will be required for both locations. And if you have further questions about that, I recommend you look at the DEX customer service on the exchange.
Very good. Our next question, and we do have quite a few coming in, so Becky, I'll have you review these as well so I make sure we're getting a good variety here. But our next question, if I make changes to my PGx test, do I have to submit another Z code? Do all PGx tests require a Z code?
Right. So again, you do not have to submit for another Z code because you just have to update it in the DEX exchange. But yes, all PGx tests in the MolDX MAC do require Z codes, just as all other laboratory developed molecular diagnostic tests require a Z code within that MAC. And that includes single and multi-gene panels.
Very good. Now, Becky, I'm going to give you a moment to review our next questions coming in. Which one would you like me to ask next?
Sure. How will the FDA's current thinking on LDTs affect PGx testing? That's probably the $1 million question, right? It's a question that everybody's looking for an answer for. The audience, I'm sure, is very well aware that the FDA has proposed that they're going to require their approval for all laboratory developed tests. And so for now, I can say that PGx tests will not be excluded from that list. As we see it now, PGx tests will be included in that process. What I will say, and this is my personal opinion, is that if your lab has passed the technical assessment with MolDX, you've proven your test analytical validity, clinical validity, and clinical utility. Proving that again to the FDA will not be as difficult. So that's just my personal feelings on, how you might prepare for those changes. Okay, how about, are any clinical trials being conducted to validate and make PGx testing universally accepted and ubiquitous? So I'd say yes. Clinical trials are being conducted. Mostly what's held PGx back from being a standard of care thus far with payers is the lack of clinical utility research evidence for drug gene pairs. So in the last, I'd say 5 years, researchers have increased research in the clinical utility of PGx. Because if you think about it from a payer's perspective, they really need to know that by testing a patient who's in their intended patient population, they really want to see that test is going to affect positive outcomes or be clinically useful for their patients. So yes, more research is ongoing, and that will help. A payer reimbursement will help the testing become a standard of care.
Very good. And I think we have time for one more question.
Yes. How about where might one find the regulation for the Novitas jurisdiction? That's a good question as well. If you recall in the slides where I was referring to the not managed care, but the Medicare area contractors, if you go to that website for CMS where they have all Medicare coverage decision information, I don't have it memorized off the top of my head, but you can search for Novitas within the CMS.com website, and you can pull up the PGx references for Novitas specifically. And if you have any questions, just feel free to contact me afterwards and I can get you that information directly.
Very good. Becky, thank you so much for your informative presentation, just sharing all of your valuable insight with us. Do you have any final comments for our audience?
Again, I'd like to thank everybody for joining today. And if you would like to get in touch with me afterwards to learn how I can help you and your PGx laboratory, feel free to reach out to me. You can reach out to Becky Winslow at inGENEiousRx.com. And I'll spell that for you because inGENEious is spelled a little differently. That's inGENEiiousRx.com. Thank you.
Becky, thank you again. And I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I also want to thank our audience for joining us today and for their interesting questions. There were a few questions we weren't able to answer, and those that are submitted during the on-demand period, they will be addressed by Becky via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Upon conclusion of this webinar, a survey will pop up and your feedback is greatly appreciated. To all our friends out there, thank you for joining us. Becky, thank you again. It's always a pleasure. Have a great day, everyone. Bye-bye.